Thanks again, and I'm sorry, unfortunately, because I was at another conference the last couple of days, I missed a lot of the content, but this seems like it was a very exciting set of talks, and the ones I heard today were really great. And so, unfortunately, I won't be telling you much about proteins, but there will be a little bit of vision at the end about how we're thinking about using some of these barcoding strategies that have been so successful in gene expression analysis to really start to understand the quantification of proteins in tissues. And I'll just kind of frame it. I'm a psychiatrist, so I frame things in the context of the brain. A lot of the things that we're developing on the side of genomics technology development is really geared towards understanding the pathology of brain diseases. And that's because of this unfortunate pairing, which is that a long time ago we thought that the brain had 3 cells. We actually thought that the ventricles, which is where the CSF lives, were the sort of important sources of consciousness, and that the imbalance of 4 humors caused disease. But unfortunately, today, despite knowing a lot more about the brain, the fact that there are hundreds of millions, or actually 170 billion cells, we still have a very simplistic model of psychiatric illness. So that really is the sort of main problem that I'm hopeful to try to tackle with developing new tools to really understand the brain in greater detail. And the actual physical study of brain tissue has been very important in neuropathology. There's obviously great examples from neurological diseases where accumulation of pathological proteins, aggregated proteins, are known to be correlated, though we don't really understand their pathological relationship. There's another one that I like a lot from psychiatry, which is this disease called general paresis, which accounted for about 25% of all people who were hospitalized in inpatient units in 1920. People didn't really know what it was. A lot of the, in the sort of earlier part of the modern era, it had a lot of similarities phenotypically to schizophrenia. But then this guy started actually looking at brain tissue from people who had this disease and realized that these people had these sort of circle lesions, and there were some interesting epidemiology suggesting it might be infectious. And then finally, we found out that there was syphilis in there because we had a stain for syphilis that was developed, and then Hideo Noguchi deployed it so we could actually see the tissue. See the organisms in the tissue. So the point is that, you know, with new stains, new approaches, that we can start to actually make better measurements and understand a pathological process in a way that actually leads to meaningful treatments. So where are we at with sort of understanding brains or tissues in general, and what can genomics potentially teach us that we don't know already? One thing I think that we're all very excited about is potentially characterizing dysfunctional cell states. So the greatest example that we're thinking about a lot in single cell analysis right now in the brain is Alzheimer's disease, because the human genetics has very clearly pinpointed microglia as an important cell type in this process, because almost every single gene that comes out of the genome association studies for Alzheimer's disease is expressed in microglia, and about 40% are specific for microglia. So we would really like to understand what are the processes that are going on in that cell type that are different in individuals with this disease. We'd also like to be able to quantify subpopulation abundances, because we know that many diseases are very specific to certain kinds of cell types, like Parkinson's disease, where you see a degeneration of specific cell types. And we also want to be able to connect disease states to histology. So we have, you know, for example, in the disease I just described, very clear lesions that we've known about for a long time. We'd like to understand how cell types and cell states are affected by the presence or absence of these particular lesions. And then from a sort of more aspirational, longer-term technology development side, we're very interested in just making other kinds of measurements of tissue beyond just nucleic acids. And the things that we're sort of interested in the brain in particular are how can we deploy some of the barcoding strategies we've developed to measure synaptic connectivity between cells, and also potentially how can we relate gene expression and other genomic features to features about anatomical structure. So I got interested in sort of thinking about these problems in general through, as Nikolai mentioned, the development of DROP-seq, which is a technology for high-throughput single-cell gene expression analysis, and the principles behind it you'll hear a little bit about in the development of this other technology that I'll describe. But the main principle is that we're using droplets to partition cells into partition single cells into tiny reaction chambers, and we're combining that with barcoded beads so that we can transfer RNA to the surface of these barcoded beads. And this particular sort of experimental process has now been adopted and modified by a variety of other groups. It was co-published with another very similar technology called InDrop from Alon Klein and Mark Kirshner, and it's really become quite ubiquitous in many aspects of biology now, because the ability to measure gene expression in individual cells, actually, as Alon said to me once, well, it's not quite as good as proteins, but it's pretty useful, right? It's sort of the first pass on a lot of this stuff. So my lab started about 2 years ago. We focus on 3 main areas. We develop new technologies, which I'll talk about. We also contribute to large-scale brain atlasing projects to kind of understand cellular diversity in the brain. Let's just get a sense of how molecular specialization really can, sort of how does it play out across the brain. There's NIH initiatives to do this. Do you want me to put the microphone on? Okay, sure. We're interested in deploying these technologies to do the pathology that I described. And so I'll tell you about 2 projects in the lab. I'll focus particularly on Slide-seq, but I also wanted to mention this other computational algorithm, Liger, because I think there's aspects of it that may be useful for those of you who are thinking about large data and other modalities like proteomics as well, which we've developed to really understand, similar to the kinds of problems that Peter probably described, to sort of integrate data sets that are quite heterogeneous from different sources that can be useful for really making meaningful comparisons across samples. So I'll start with Slide-seq, and I want to mention that this project is a very equal collaboration with Fei Chen's lab. Fei is the developer of expansion microscopy in Ed Boyden's lab, and brings just an incredible talent for imaging that and sort of combining that with really sophisticated ideas in genomics. And so we've just had a terrific collaboration, and our students, Sam Rodriguez in his lab and Bob Stickles in my lab, co-developed the technology together. So Slide-seq really comes out of identifying sort of 2 really critical needs in our single cell analysis. The first comes from these atlasing efforts I mentioned, where we're getting gene expression patterns. Yuval, I think, mentioned a lot about T-distributed stochastic neighbor embedding. This is one such plot of a relatively small piece of the mouse brain. And you can see that there's just an incredible amount of diversity within this structure, sort of on a neuron, sort of total number of cells to total number of biologically meaningful clusters. I think it's one of the most diverse that we've ever sampled. And I've circled that region of the mouse brain here. And anatomists have previously noticed that all of these little substructures that are drawn in these light lines have very different projection patterns and different sort of biological importances. So for example, there's this little tiny structure here. Someone found a creline by accident that happened to drive in that substructure. And if you excite those cells, the animal gets super nervous. And there's this other structure over here where the animal gets very hungry, right? So there's just really, really very fine specialization. And as we're trying to understand how these gene expression patterns relate to this particularly complex structure, it's very hard to think about how you could do that in an easy, systematic way with designing probes, because there's just a lot of genes here, and it's hard to sort of design enough experiments to do all the right combinations to really systematically map cells into space. And so just being able to locate genes in space in a very sort of whole transcriptome, unbiased fashion might help us to solve this problem of spatial mapping of cell types. The second thing that I've already mentioned is the interest in relating gene expression and cell states to specific lesions. So for example, I've shown this nice, beautiful stain of an amyloid plaque from a recent paper by Ado Amit, and different cells that are infiltrating around this plaque. It would be nice to know what their gene expression patterns are, and we could do some interesting computation to sort of relate these cell states to their proximity to amyloid plaques or other lesions. And similarly, different diseases like these different prostate cancers have different prognoses, but they also they have strong relationships between their histological architectures and those prognoses. So understanding molecular cell types that are present or absent from those particular structures would be very useful. So how do we do this in nucleic acid chemistry? The main class there's 2 sort of main classes of approaches that people take.
One is in situ hybridization, and we've already heard some really terrific work from Sarah describing how you can really see and visualize individual transcripts with very high sensitivity. There are now also some interesting approaches like MIRFISH and StarMap and SeqFish that allow you to multiplex these measurements by doing some very clever combinatorial indexing and sort of repetitive probing of these samples. The advantage of this is that these approaches tend to be incredibly sensitive, and that you get very high resolution, single molecule resolution in many cases, depending on the abundance of the transcripts. The downside of some of the approaches is that, first of all, they require a lot of expertise. So many of these technologies are very hard to export to sort of routine molecular biology labs. And the other thing is that it's hard to generate very large volumes of tissue or large numbers of samples. And finally, another challenge can be that you have to identify up front the genes that you want to probe. So sometimes you may not know whether a gene is important in space up front. The other sort of class of technologies is one that just particularly is embodied by this technology called Spatial Transcriptomics from a group in Sweden, where they spot barcoded primers onto a surface of a slide and then recover RNA from that from a tissue sample that's laid over it, and they use that barcoding to then sequence the resulting cDNAs and recover their positions in space. Those dots I'm showing here are rather large. They're about 100 to 200 microns. And that can make it very challenging to find important features. For example, in the little sample of tissue I showed of the bed nucleus, most of those structures are much smaller than 200 microns. So we kind of wanted to develop a technology that would sit in the middle here, something that gives you the sort of unbiased capture of gene expression like spatial transcriptomics, and also the ease of use of spatial transcriptomics, because you don't need a microscope to do this. If you know the coordinates of the barcodes, you just have to submit it for next generation sequencing and you get the image out just like that. But also had a little bit more of the resolution that's afforded by the in situ hybridization approaches. So this is the scheme that we conceived of for Slide-seq. We take the beads that we developed for Drop-seq. The Drop-seq beads are beads that have a large amount of a single barcoded primer on their surface. So it's an oligo-TT capture to capture mature polyadenylated RNAs. And that poly-T primer has a barcode that's different from the barcode on every other bead. We scaled down the size of these beads that we use for the synthesis down to 10 microns. And that's what gives us our feature size. And then Bob and Sam figured out a way of sticking these beads on to a transparent glass microscope slide. And so you can see we've arrayed out a set of 15 of them here. And then if you zoom in on any individual one, you see that there's about something like 60,000 of these spots on an individual 3 millimeter disk. And if you zoom in even further and you look at individual beads, you see that they're nicely packed, and they're quite porous, and that's what allows you to get a lot of primers on the surface and capture a fair amount of RNA. So we make these slides, we call them pucks, and then we sequence the bead barcodes on these pucks to identify the physical location of each one, because each barcode is random. We have 15 bases of barcode sequence here, so we have 4 to the 15th unique sequences. So this is a round of the sequencing that Sam and Fei developed to identify the sequences at individual locations. We use an old technology called SOLID, which actually has been discontinued, which has caused us some headaches as we're trying to scale this up. But there are other approaches we found that work subsequently. And so the idea here is that this imaging and indexing process can be done up front, and you can accrue many, many hundreds of these slides if you like. You store them in the fridge. And then and you can also store on just a 2-column matrix of the XY coordinates of these beads. And then you can come back and do your experiment where you want to take a piece of tissue and put it down onto the slide. And that's a process we developed to basically transfer the RNA to this bead surface. And then you basically are able to, after a small number of steps, take this whole slide and stick it into a Eppendorf tube and just do molecular biology as you would for a bulk sample. And so it's quite straightforward 3-hour process to do. So to show that this actually worked, we went to the hippocampus in the brain, because the hippocampus has a very stereotyped architecture that I'll show in a minute. And so here what I'm showing is the 60,000 beads from an individual sample that we've clustered as if they're single cells. So this is just a t-SNE representation of these individual beads. And then we've done some clustering, and we've identified individual populations, and we can annotate those populations by differential expression. The genes that are markers for these populations are genes we recognize as cell types that we've seen in our single cell data. But we don't just have this t-SNE representation of data, we also have a spatial representation of the data. So if we map those clusters that we've generated onto their physical locations, we can see that we recover a lot of the really nice spatial features that you'd expect to find in the hippocampus. So again, this is not done by microscopy. There's microscopy just to index the locations of the beads up front, but we now are able to recover these spatial features just by clustering based on the fact that many cell types have certain spatial relationships with respect to each other. So the statistics behind, sort of the stats behind what we're able to capture, we can capture about 300 unique transcripts per cell at a spatial resolution of 10 microns, and I'll show you some further validation of that in a minute. We sequence these individual 3 millimeter pucks for about 200 million 200 million Illumina reads. And then what's nice is that these experiments are very straightforward. So one can do about 100 of them in a day without really it being extremely strenuous. So for those who are not familiar with the hippocampal structure, architecture, this is just a DAPI stain of this tissue. And you can see that, again, we have a really nice recapitulation of even some of the subtler architectural features. This kind of midline V feature of the CA3, for example, you can really see nicely in our Slide-seq data. And I'm zooming here just to show you another one of these fine features. We can recover this blue layer of ependymal cells that really lines the ventricles, again, the CSF cavity of the brain. That's also flanked by these two yellow dots, which are the habenuli. It's another structure that's a subcortical structure. So we wanted to quantify, again, the diffusion, make sure that we weren't really smearing. I mean, it seemed pretty clear that we were getting pretty nice recapitulation of structures. We wanted to make sure that we wanted to measure it in a little bit more of a rigorous fashion. So what we did is we took one of these hippocampal slices, and we looked at the Slide-seq data and just basically measured the thickness of this CA1 field feature, which is the granular layer. And the granular layer comes out just by looking at the total number of transcripts, because there's a lot of cells there. So you can see that little line there just because there's a lot of cells and you're capturing a lot of RNA. So it's essentially like a DAPI stain. And then we just compared that thickness to the DAPI stain here, as well as the stain for two other genes that are pretty abundant in that layer. And what we found is when we looked at, sort of comparing the full width half maxima of each one of these individual probings, we found that the thickness was pretty much the same, and that the resolution, we believe, is largely driven by the thickness of the size of the bead. So one thing we're doing now is we've just bought a oligonucleotide synthesizer, because we did a lot of this through a company called ChemGenes. But now we bought a synthesizer that is capable of synthesizing on substrates that are far smaller. So we hope to be able to reduce the feature size down to, let's say, 3 microns or something on that order. This seems to work pretty well in other tissues as well. Here we've just done the same kind of clustering that we did in the hippocampus, but on the liver. If we look at two of the individual clusters that emerge, they clearly represent features of gene expression that are known to distinguish periportal from pericentral hepatocytes. And we can also identify individual individual nephron substructures like the glomeruli, proximal and distal tubules. Oh, thought somebody said something. So what about this critical need we identified to map cell types from gene expression into space? So one way of doing this, which is simple, is to take the markers for these individual populations, for example, and just see if we can identify specific spatial localizations. So we did that in hippocampus data. We took our single cell data from the same structure. We found a bunch of genes that are quite unique to these different populations, and we just mapped them in space. And we can see that the accumulation of these signals seem to be very nicely recapitulating the locations that we knew, because these cells have been studied before, so we knew where they would locate. One thing to point out that's kind of nice is this neurogenesis population is one of the few parts of the brain that continue to regenerate in the adult animal. And these neurogenic populations are actually found in this inner leaflet of the dentate. So you can, again, see the kind of fine resolution where you're seeing it very much accumulating in that layer as opposed to the kind of outer part of the dentate gyrus. So that particular slide was one of several, about 66, that we generated from the same animal. So this is a 3D reconstruction. And we're coloring the beads now by just 3 different identities, the CA1 slash CA2 fields, the CA3 fields, and the dentate gyrus. So you get a sense of the ability of this technology to scale to large 3-dimensional volumes. Here are just a few examples of the individual data points that we generated, the individual slides. So we also were thinking sort of analytically about more kind of formal ways of mapping cell types into space, not just picking some markers and seeing how they looked. So Alex Gueva in the lab is a statistician. She developed an approach based on non-negative matrix factorization. To just briefly to describe this approach, she took the single cell data and she factored it with NMF. The single cell data already had, from this particular tissue, which is the cerebellum, already had labels associated with them, cell type labels. So she looked at the factors that came out of this NMF factorization, which is kind of like PCA. It's a way of reducing the dimensionality and finding metagenes. And she identified that certain metagenes correspond to certain cell types. And so she created a dictionary of metagenes versus cell types. And she used that dictionary to basically regress the Slide-seq data using non-negative least squares into space. So a few examples of this were the granular cells, which are occupying a sort of thick portion of the cerebellar cortex. And you can see that being recapitulated nicely with this projection. The Purkinje cells, which are a thinner layer of single cell, but they're quite large, outlining just above the granular cells. And then some even rarer populations we were able to pull out include the unipolar brush cells. These are a rare interneuron population that accumulate in specific regions of the cerebellum. So this one here you can see is called the nodulus, and you can see kind of an overabundance of these unipolar brush cells there. So the other need I mentioned was this sort of the ability to link gene expression to tissue pathology. We have more to go with this, but I want to give one example that Sam developed, which I think is kind of interesting. And this is a puncture wound model. It's a traumatic brain injury model of the brain. And the reason that Sam was interested in doing Slide-seq on this model is because this is actually a experimental process that's done by almost every neuroscience lab, which is called stereotactic injection. If you want to go in deep into the brain to deliver a virus or a pharmacological agent, you have to use a needle, and you go through overlying usually cortical tissue. And there's sort of a presumption that after a couple weeks, the brain kind of heals and everything's okay enough that you can, do your behavior or your analysis at that time. So Sam had been doing a lot of these experiments for other things, and he was just kind of curious, like, what happens to the brain over time? Is 2 weeks actually a good amount of time? What happens to this area that's kind of going to get scarred over? So he did a series of kind of mock injections where he just stuck a needle in that had saline in it and then removed it. And here's an example of one such injection where or faux injection, where he penetrated the thickness of the cortex. And this gene VIM is known to be upregulated in response to puncture injury, or mechanical injury of the brain. So after 2 weeks, after 1 week, you can see this upregulation of these presumably glia that are kind of being reactive in this location. We're able to kind of map the location of this puncture wound using just DAPI staining, because there's a kind of accumulation of cells that kind of breaks the regular architecture of the cortex. So a couple things we can do that are sort of interesting. One is we can take the same approach that Alex developed to project cell types into space and start to measure where they're accumulating around a lesion. So here, for example, we're looking at the accumulation of fibroblasts right in the location of the injection site. And then a thicker sort of layer, a set of layers of microglia kind of accumulate around the area as well. This also recapitulates known work about sort of what happens in the context of a mechanical injury to the brain. But one thing that we were really surprised by was that after 2 weeks, we noticed that in the areas around the wound, there continued to be an upregulation of immediate early genes like FOS and JUN, which are known in the brain to correspond to hyperactivity. So this really suggests, and we saw this continuously in multiple replicates, this suggests to us that the brain continues to not be happy with this injection event, even many weeks after, and it may make us think a little bit more about the controls we need to do when we're doing these kinds of stereotactic injections in neuroscience. And then finally, I wanted to just highlight one other application of the work from the same cerebellum analysis I mentioned a little bit from the projection of cell types, which was to basically just use this technology to kind of GPS localize genes in a specific structure and start to pull out patterns that we may not have appreciated before. And this is because it's a little bit of kind of heavy neuroscience. I'll try to go through it a little bit as sort of briefly and cleanly as I can. But the cerebellum has this layer, which is the Purkinje layer that I've traced out here from one of the Slide-seq experiments. And it's known that they come in two flavors, the zebrin positive and zebrin negatives. It's called zebrin because this molecular distinction, which was found just by an antibody, no one knew the target actually for a while. It turns out to be the gene ALDOC. This pattern looks like a zebra if you look at it sort of over the longitudinal surface of the cerebellum. And so what's interesting is that despite knowing about this for a long time, we don't really know sort of the compendium of all genes that show this pattern. We also don't know if there are other patterns within this cell type that we might not have appreciated before. And so Sam developed some nice algorithms that allowed us to really quantify this and look in a kind of unbiased way across the transcriptome to find new genes that have interesting spatial localizations in the cerebellum. And there are a few different things here, but I thought I just would highlight one, which is this gene MYBPC1 that he found that has a very similar pattern to ALDOC, but what's kind of interesting about it is that it wasn't in the Purkinje cells, it was in the glia that are next to them. And we confirmed this using both the Allen Brain Atlas, which is this wonderful resource of gene expression patterns that have been done for every gene across the whole genome in the mouse brain. And we also confirmed it with single cell data that this gene is very specific to Bergman glia. So it's the first example of this pattern occurring in a non-Purkinje cell population. So, and it's also something that we wouldn't have identified just from the single cell data, presumably because there just aren't enough genes that have this pattern that they come out in an unbiased analysis. So I think it sort of underscores the value of just sort of a spatial analysis of sort of the whole transcriptome. So at a glance, Slide-seq, again, affords you about 10 micron resolution gene expression. It's done on fresh frozen tissue. It's untargeted across the whole genome, but its capture efficiency is limited. It's considerably more limited than the capture efficiency currently for single-cell approaches, like 10x genomics or DropSeq. And what we like about it is it's quite scalable to building large three-dimensional volumes of tissue. We think it'll be useful for things like profiling cancers, for example, where you don't know what the architecture is going to be because every cancer is quite different. And again, discovery of spatial patterns like the ones I mentioned in the cerebellum, we think is one particular area where we think it'll be immediately applicable. And then I wanted to just mention some future technology development that may be of interest to some people in this group. We're very interested in thinking about how to deploy this in non-fresh frozen tissues. So can we use archival brain tissues that are fixed or FFPE samples? And some of that is also related to interest in targeting particular genes that will also give us greater read sequencing efficiency to identify specific genes, not just the whole transcriptome. That can be useful for some experiments. And then Fei in particular is very interested in extending this to multimodal analyses. And one of the most immediate applications we can see is the fact that we can now barcode antibodies, and this will allow us to potentially co-stain for many, many different proteins and use the unique nucleic acids that are bound to those antibodies to simultaneously read out protein and gene expression at the same time. So in the time I have, I just wanted to also talk about this other project, which is really about taking single cell analysis, not Slide-seq data at this time, although we have started to use this approach with Slide-seq data, to really start to really compare and contrast data sets to really reveal interesting biology with these single cell approaches. And so the reason that we're we're really interested in this is because I think we've kind of started to reach the point where everyone recognizes that single cell analysis with these very large data sets is useful for figuring out all the different cell types that might be present in a tissue, right? That's something that's pretty easy. A within data set comparison, we do pretty well at. But a lot of the problems that we're really interested in are comparing cases to controls, diseases to other diseases, or to normal people. And those comparisons can be very tricky because there are batch effects, as we've heard about before, but there can also be other confounders that I'll mention in a minute. But what are the kinds of things we want to compare? We want to look at individual variation, the kinds I already described. There's also interest in doing cross-species analyses. You know, we have a lot of models of diseases, but those models sort of presuppose that the mouse version of a particular brain structure, for example, is a good correlate of the human. It'd be nice to know that in a kind of quantitative way. And the other thing that's really driving a lot of this work is the fact that there's a lot of new technologies that are being developed, and we'd like to be able to integrate those technologies together. So I'm giving you a sort of a summary of the omics data that people are generating from very, very large numbers of cells at this point. And there's a lot of very mature single-cell RNA-seq approaches that give you, thousands to millions now of cells. Now we are starting to infiltrate epigenetics, so we can measure, the chromatin accessibility and methylation of individual loci from individual cells. And also this explosion that Slide-seq is participating in situ analyses. So there's such a rich opportunity to be able to unite these kinds of data sets together, and it's challenging. There's sort of what we might think of as batch effects, like for example, if I wanted to integrate drop-seq data with Mars-seq data, those have challenges because these different technologies have different biases. But there's even greater and but also more exciting opportunities if you can kind of go across these modalities. But this is hard because these data have very different distributions. I'm just showing some examples here where mean and variance of methylation and RNA data are very different, so you can't just treat them as equal. That's kind of obvious. They're actually inverted from each other. Methylation usually is inversely related to gene expression. In the case of one of these in situ techniques I mentioned, StarMap, there's actually a very high false positive rate, or not high, but that's its failure mode. And in drop-seq, the failure mode, or the single-cell data sets, the failure mode is high false negative. So that creates a lot of differences in your data that's hard to reconcile. And of course, if you want to go cross-species, you see a lot of noise, right? Just hundreds of millions of years of evolution. So if you were to just use sort of standard PCA and t-SNE to look at these data sets, you would find that they completely separate by their sort of modality or data set type. And this is also the case for many comparisons within RNA-seq, if you look at one technology to another. And so I was lucky to have Josh Welch join my lab right as I started. He's a very talented computer scientist who also had experience with single-cell analysis as a graduate student. And he adapted a scheme that had already been published for doing integrative analysis in bulk data, and he figured out a way of applying it to single-cell data. And the main engine of this is this modified form of non-negative matrix factorization called INMF. And what this INMF algorithm does is it takes a bunch of data sets. Here I've shown 2, but it can use as many as you want, N1, N2, N3. And instead of just sort of looking at a low-dimensional representation of the data, H by W matrix multiplication, it tries to find 2 sets of metagenes. One that's shared, that's the black W matrix, and one that's data set specific. And the optimization function has a parameterization to kind of weight the penalty that you weight on the data set specific factors. So you try to basically lower your lambda as much as you can while simultaneously maintaining alignment. That's sort of the overall principle that we're doing. And the nice thing about this integrative strategy is that it allows you to look at what's shared and what's different about individual data sets. It emerges just de novo in an interpretable way from the analysis. And then we came up with a clustering strategy that we needed because these data sets still exist on different scales, and we had to figure out a way of integrating them together beyond just this kind of comparing and contrasting analysis. So that we call this a shared factor neighborhood graph. You can look at the paper if you're interested. It came out last week in Cell. So we used a set of data sets a pair of data sets that have been used sort of for batch correction analysis in this space of single-cell analysis. They're peripheral blood cells that come from 2 different technologies of single-cell analysis. If you use them, analyze them together as if they were the same data set, they would separate, again, just like the other ones I showed. And our approach, which we call LIGER, does a really nice job of integrating these data sets together. There's another approach from Rahul Satij's lab implemented in his package called Seurat, and our approaches seem to do a pretty good job of aligning these data sets together. So if we quantify that just by looking at the nearest neighbors of any individual data point here and seeing how many come from one data set versus another, we see that we're both obtaining relatively similar alignments. But one thing we liked about our approach is that we also quantified something called agreement, which is how well does the neighborhood that this cell experiences in this joint analysis compare to when you analyze the data sets separately, because this is basically about how much information you're losing when you integrate cells together. You could start to blur the lines. And we found that LIGER did a considerably better job of this. And this can be sort of pushed to its extreme if you take 2 cell types that are very different from each other, that should never align. So those are these 2 here from the brain, neurons and oligodendrocytes. They should never align. And you can see that we continue to recover a lot of the substructure that can be lost in other integrative approaches. And so that's shown here quantitatively. Yeah?
What is the scale of the agreement metric? What does 0.2 mean?
Yes, it means that within a neighborhood of, I believe it's 25 nearest neighbors, how many nearest neighbors are shared between the original analysis and the new analysis?
So it's a fraction of shared neighbors
Yes.
between the 2.
Yeah.
Thank you.
Yeah, sure. So how can we apply LIGER in kind of biologically useful ways? So one thing we did is we wanted to sort of show was that when we get new data, we can go back to old data sets from single cell and we can find interesting things with them, even if those data sets were prepared in very different ways. So here's an example of this analysis that I showed you initially of the bed nucleus, this interesting very interesting heterogeneous substructure in the brain. And one of the populations there we flagged just because the biology seemed interesting. It was expressing a gene called VIP, which is known to be expressed in a subset of neurons in another region of the brain, the cortex. And we were curious, is this just one marker or are these cells really the same? And so we used LIGER from this old data set that was published when I was in Steve's lab. Again, different technology. These are actually nuclei that we're profiling versus cells, so there's a lot of differences here between these 2 data sets. And when we integrate the data sets together with LIGER, we find that a subset of the interneurons from the cortex that we thought might be related to these cells actually do align. And they share a lot of markers, all the markers that the folks who study these cells would say that defines these cells. So we think that we've found the fact that these cells may have, there be a subset of these cells may migrate to this structure, something that people hadn't identified before. And again, it was just by kind of, it's almost like a blast, right? You're blasting one new data set that you've generated with a set of old cell types that you found from some other published data set. We're also really excited to deploy this to the human brain and to do kind of pathology of the human brain. We're very excited and we're quite fortunate that when you freeze postmortem human samples, the nucleus somehow does a very good job of protecting RNA. So we can get very, very, very high quality RNA, and many labs have shown this, from individual nuclei in the brain. Thousands of genes detected per sample from postmortem human samples. And they seem relatively resistant to postmortem interval and all the things that people often agonize about when using cadaverous tissue. But again, there's still batch effects, and so if you do standard analysis, you see that cells separate. These are 7 different people. You can see the cells are separating by people. And so LIGER does a nice job of integrating these data sets together, and you can see that the individual cell types you'd expect to see in the structure of the brain, the substantia nigra, we're getting kind of nice expression of these specific markers that we'd expect to see. Again, I mentioned this ability to compare and contrast, and I wanted to just give an example of how this can be useful for pathology. So here I have 7 people that have all been told by me by the brain bank that they're normal. But we flagged the fact that some of these people had relatively high amounts of the data set specific factors that I mentioned compared to other people. So when we looked at these people and their particular factors, we found that some of them, one of them, for example, expressed a lot of immediate early genes. These are genes that are involved in stress and injury. And again, I mentioned those in the Slide-seq portion of the talk. Turns out that this particular person died in a car accident. and had a documentation of a contusion. So probably they had some kind of brain injury as a result of death, even though they were normal up until the time of death. And that was the reason they were given to us as a control. And then this other person who I'm highlighting here, so these plots show the genes that are highly loading on a specific person versus the ones that are loading on a shared across people. This other person, 5840, in their microglia have a bunch of genes that are known to be involved in reactions to amyloid accumulation. And it turned out that when we went back to the brain bank, even though this person had no history of dementia, it turned out that they incidentally were found to have amyloidosis in their brain. So we think this is a nice way of being able to pull out meaningful genes in these individual cell types and really sort of do some kind of pathological analysis just with the factorization that we've done. Final thing I wanted to mention before wrapping up is an integrative analysis that Josh was able to do between methylation data and gene expression data. There's a lot of really cool opportunities you could imagine with doing these kind of integrative analyses to really understand gene regulation. And this is a pretty challenging analysis. Methylation, again, goes in the opposite direction. More methylation gives you less gene expression, so you have to reverse the sign of the data. There's also very, very different throughputs that are possible with methylation data. So Joe Ecker's lab is like the world leader in this, and this is a published data set, pretty much the largest data set available, and it's 3,000 cells compared to the fact that we're routinely slinging around millions of cells now in single-cell gene expression analysis. So Josh was able to integrate 50,000 drop-seq cells from the same frontal cortex region of the brain with these 3,000 single-cell DNA methylation profiles. And we generate these river plots that show you the correlations between the published clustering of the gene expression and the methylation with the shared clustering. And you can see that because there's very thick and sort of uniform lines between clusters, that we really think these clusters are biologically meaningful. They're capturing the shared cell types across these two very different modalities of data. The nice thing about having these two things paired together is you can now look at gene regulation. So we can now say which elements, intergenic elements, let's say flanking elements of the ARX gene, I'm just giving this as an example of one that Josh found that was kind of cool, which one of those particular elements is correlated with gene expression across many different cell types in the cortex. And he identified these individual elements. They turned out to be quite conserved sequence-wise, which tells you that they might be important for gene regulation. And in fact, a couple of them were actually shown. The reason we look at this particular locus is because we knew that there were some that had cell type-specific regulatory implications. And so we found the published examples of this and also some other regulatory elements as well. This could be useful in a lot of different settings in human genetics now. We're trying to understand the when we look at an association study across disease, we might identify a gene. We don't necessarily know the functional consequences of these individual loci we're identifying. This could be a way of flagging particular regions that are near to an association as being important for regulating the expression of the nearby gene. So in conclusion, we really think LIGR is broadly useful for doing analyses across individuals. I mentioned making comparisons in postmortem tissue and also now looking across modalities. We, in our paper, also did analyses with spatial data, the STAR map data set from Carl Dijsselroth's lab. It also works when integrating with Slide-seq data as well. And we really think it scales well to the kind of large data sets that we're getting now in nucleic acid biology or nucleic acid genomics. And we're really excited. We're going to be using this now. We are using this now to really start to do pathology in the brain, and this is really what I really wanted to do when setting up the lab. And so here's an example of some new work from a student in my lab, Tushar Kamath, and RA Abdul. And they've been able to profile nuclei from a bunch of different human donors and from mouse donors and isolate specifically the cells that die in Parkinson's disease from this structure, the substantia nigra, and actually have found a population using LIGR that seems to be very unique to humans. And so we're really following up on this and seeing whether we can understand whether this population is differentially sensitive to the Parkinson's degeneration process. So with that, I think I'll just, again, recognize the folks who really led the work that I described, especially Fei Chen, the terrific collaboration that was led by Bob and Sam, and also Josh, who actually now has his own lab at University of Michigan, who's really spearheading a lot of new algorithms in single cell. So thanks very much, and happy to take any questions.
Marco has a question.
Hi. The first thing I might have missed, and it might simply be too simple, but I guess you integrated Slide-seq already with like full single-cell sequencing data to then, because you could use the same things like Surat and so on to then basically map the whole, how well does this work?
Yeah, so the approach that we use in the paper is basically just defining the space with single cell data and then just projecting the space.
Exactly.
So we don't use the spatial data to contribute. We do see somewhat similar results when we use Liger or other approaches that are integrative, where the two data sets would participate equally, or you could maybe even weight those. But we find that the spatial data is so sparse that it can be very difficult to integrate and have them participate equally.
Okay. And the second question goes into this direction. So because of the sparsity of the data, when does something like Liger basically fall off, you know? Because if you only look at, first you try to find the common genes and then the specific ones, but you cannot separate it, you not sequence something because it's really not expressed and so on. So just about the principle, in the end, on how many genes contribute mainly to the, to the partition?
To the partition with respect to identifying cell types in space, exactly, for example? Yeah. That's a good question. I mean, I think, and I guess probably the answer is that NMF in many ways forces a relatively sparse representation of cell types. So even if you look in single cell data, NMF, if you look at sort of the loadings of individual genes on a particular metagene, it can only be maybe 10 to 50 genes overall anyway. And I guess part of that is the fact that gene expression is so correlated that you don't need that many individual genes to represent things. But I'm not sure if, so most of my familiarity with is projecting the data into Slide-seq into Slide-seq data. And I think that almost every gene that is defined by the single cell data participates, because it's not like they're all 0 or something in the single cell data, it's just much sparser. So I think they all participate, but on an individual cell by cell level, it's undoubtedly less than it is on an individual cell by cell level with respect to the 10x or single cell data, if that makes sense.
I have a question. I wonder what are some of the factors that contribute to the lower capture efficiency of Slide-seq, and if you have some ideas of how you might alleviate these.
Yeah, that's our number one sort of focus right now. The one major thing we know is that there is a kind of relationship between tissue permeability and capture. So we're very careful. We wanted our resolution to be very high because that was important to us. But we know that if we add additional detergents or denaturants, we can push the capture efficiency considerably. But at the same time, we don't want to lose the resolution. So that's the thing we're working on right now. We have found a way in that regime of increasing the efficiency by about 50% over the published work, which is encouraging, but I think there's a lot more that we could be getting out. The other thing that I think is happening is we only have a single surface that's capturing off of tissue, right? So basically what I think is happening, and the reason that the diffusion is so limited, is that you basically have, an RNA kind of makes a choice. It's diffusing, and it can either diffuse towards the beads or away. And probably if it diffuses away, it's never going to get to any beads. It's just going to go out into solution, right? So we probably lose a lot of RNA because it just never actually migrates towards the surface. And so the most logical thing to do next is to try to drive stuff into the surface of the beads. And there are, we can go back to, Edwin Northern to kind of, see or Southern, I can't believe I called him Northern, but to, sort of think about ways of transferring RNA onto surfaces. Yeah.
Cool, thank you.
Yeah.
Luca has a question.
I hope this is not too naive questions, but it seems to me that at the moment you're focusing on the genes that allow you to, all the transcripts that allow you to differentiate between the type of cells. Is there in the data also some indication of what are the temporal dynamics of gene expression, more like in the function? So is there anything that you're thinking in this direction?
That's a great question. So even in the single cell data, we definitely see things like circadian patterns. We've seen that before. it doesn't quite map on exactly to what you'd expect from kind of the canonical literature, but some of those genes do show interesting variations across these populations. I think there is a lot of rich opportunity, especially in the brain, to really understand the extent to which these populations are fixed and never shifting into other populations over time, and the extent to which they might actually be dynamic. And I think, unfortunately, those experiments require, at this juncture, a lot of either sophisticated barcoding in genomics or a lot of long, arduous Cree line generation and things like that. But I actually think there's a lot of really exciting opportunities there, because you see sort of things that seem tantalizing, like patterns that you look in the literature and it may not be mentioned in the brain, but it's like this gene is in response to stress, or it's glucocorticoid sensitive. It doesn't strike you as necessarily something that would be specifically defined in development and then never changing. So I think there is a possibility that some of these populations are actually, I'm going to a conference where we're supposed to be talking next week about what's the difference between a type and a state in the brain. And I think the answer is we just don't have enough information, but I think that there are a lot of examples of things that people think of as types that are actually states.
One more question over there.
You already commented on that a little bit. For the even when you get the RNAs to diffuse in one direction potentially, do you think the density of the oligos per bead is going to be a limiting factor for the capture efficiency? Do you expect, how hard of a limit is it going to be?
It's a good question. We do expect that at some point it will be limiting, that the density on an individual bead. So for example, if we try to do drop-seq with these beads, they're only 10 microns, you cannot capture as much material as you can when you go to the drop-seq beads, which are 100-fold larger in volume. So we suspect that we're not delivering as much transcript as we could. There's a lot of caveats to that. I'm happy to discuss it in further detail, but we have a long way to go before we reach that, probably another 5-fold.
And another aspect to this actually is that as you reduce the size of the beads to gain spatial resolution, you also increase the surface area. So potentially that can actually help with the problem of providing more barcodes.
That's a very good point. And we've also thought about doing things like stacking the beads, things like that. These are great questions. Yeah.
Wonderful.
So
well,
things I think about all the time. So
questions
Just
is
one more?
one of the things that we certainly try to encourage. Yes.
I just wondered if you'd done anything with correlating moldy tissue imaging, like the kind of things that we're involved in for the maybe lipids or metabolites or other related compounds that should be there spatially, and you could see with the mass spectrometer.
The most straightforward way to do that with the technology currently is to do alternate slices, right? Of course, you don't have perfect correspondence between cells. Mm-hmm. The next best thing and I guess wouldn't necessarily be able to do things that are destructive to the tissue, but would be to do what we're excited about right now is to fix the tissue and do various kinds of non-destructive imaging prior to the process of capturing the RNA. And that would allow you potentially to profile things that couldn't do mass spec because that's destructive. But
yeah, but even in mass spec imaging, you don't really sort of dig giant holes through the tissue when you do these experiments typically, so it might still work.
Okay, that's good to know.
That you could try it with matrix on top and then see if your capture is working independently.
Yeah, no, I'd be interested to talk more about it.
Wonderful. Thank you again.
Thanks.
Evan.
Thanks.